Hi everyone. I just wanted to go over a few things before you guys take your test. I wanted to review evolution by natural selection, the 5 fingers of evolution that we learned about, and you have those in your notebook, evidence for evolution, and cladograms. So here we go. So first, evolution by natural selection. The 4 postulates that Darwin came up with to support evolution by natural selection were variation. That means that individuals of a population or individuals of a species can have different phenotypes. So we can get dogs is a good example. You know, we can have a lot of different types of dogs, but they're all in the same species. Adaptation. Adaptations can be things like an elephant's trunk is adapted to pick things up. Penguins have sharp beaks to catch fish with. These are just changes in an individual that are beneficial, so they get passed down to the next generation, helping those animals survive. Overproduction, that is when more offspring are born than will probably survive to reproductive age. So animals will have a lot of offspring because chances are, either by disease or predation or lack of food, chances are not all of those individuals will make it to reproductive age. We've also got descent with modification. So that means that that's kind of the survival of the fittest part of this, where beneficial traits are selected for, harmful traits are selected against, and the ones who have beneficial traits, the individuals who have beneficial traits, will survive and reproduce. So for example, like giraffes, if a giraffe has a long neck, it has that adaptation, it will be able to reach more leaves, it will be able to produce more offspring, feed those offspring better, and then the next population will have more long-necked giraffes than short-necked giraffes because they reproduced more. So that's evolution by natural selection, variation, adaptation, overproduction, and descent with modification. So next, let's talk about the 5 fingers of evolution. The first, remember we represented with the pinky finger, that was genetic drift, and that is basically the gene pool being simplified. So we had things like the bottleneck effect, where we have like, we talked about the forest fires in Australia being a bottleneck effect because a lot of individuals would have been taken out of the population, therefore decreasing the genetic diversity of that population. Bottleneck effect is one of them. Founder effect is another. That's when you have a large population and a few individuals. So let's say like birds on an island. If there's a large population of birds, maybe they're running out of food and a few birds decide, hey, let's fly to the neighboring island and see what's over there. Those few birds fly to the next island, they make a new population, and they have founded that new island. So that's the founder effect. That new island, however, will have decreased genetic diversity than the bigger island because only those individuals are there. So when they reproduce, they'll pass down their genes, but it will just be their genes and not all of the diversity of the previous island. So that's genetic drift. The next one is non-random mating. So we talked about mating being due to appearance and location. So appearance, a really good example is peacocks, right? The reason the male peacocks have those big colorful feathers and they put them on display is so that females will see, hey, that male has clearly a good food supply, he's healthy, so I should mate with him. That's the appearance bit. And then location is pretty simple. Individuals, organisms will mate with whoever is nearby and appeals to them. So those birds on the islands, they're going to mate with the birds on those islands. They're not going to fly back and forth to mate. It just doesn't make sense to spend that much energy getting to another location when there are individuals of your own species nearby. So that's genetic drift, non-random mating. The third one, the middle finger, was mutation. Remember that when DNA is transcripted and translated, we can get mutations in the DNA, and these are just random changes. They can be caused by nothing at all, or they can be caused by environmental factors like UV light can cause mutations in DNA. And that causes a lot of differences. That causes differences in our genotypes and our phenotypes. So that is part of evolution. So the next one is gene flow, and that is when individuals move into or out of a population. So immigration is into, emigration is out of, and gene flow can alter the allele frequencies in a population because individuals are moving back and forth, or just leaving for good or entering to stay. And the last one is adaptation again. So adaptation definitely pushes evolution along because individuals with better adaptations are going to survive and reproduce more. So genetic drift, founder effect and bottleneck effect, non-random mating based on location and appearance, mutation causes genotype and phenotype differences, gene flow, moving into or out of a population, and adaptation. Those are the 5 fingers of evolution that we went over. Those will be on your test. The last few things are evidence of evolution. So let's talk embryology. Embryology, we saw that embryos of different species actually look very, very similar very early on in their development. And then as they progress, they get their differences that make them different species. So for example, we saw that chicken and turtle embryos look really, really similar early on, but then as they grow, the turtles get shells, they get tails, the chickens grow their wings, and then they look different by the time that they are born or by the time that they hatch. So that is comparative embryology. That's a supportive evidence of evolution. The fossil record. Remember that the further you dig, the older the fossils are. So we can actually see this really cool timeline of organisms that lived at different times. And we can say, hey, this organism is buried with a bunch of shells and other sea creatures, so this organism must have been aquatic. We can kind of use logic and the timeline that the fossil record gives us, and we can see organisms develop as well. And we can also find intermediate forms of organisms. So we saw in our lessons, we saw the Myocetus, the walking whale, right? The whale that had really small hind limbs. We saw Archaeopteryx, which was the dinosaur with feathers. So we can see all these different intermediate forms that are supportive of evolution. Hi, okay, I'm teaching. Anyway, so comparative anatomy is a big one that we went over. So homologous structures have the same structure but a different function. So we talked about 1 bone, 2 bones, lots of bones, digits. So we use those bones for grasping, but other organisms use them for a lot of different functions. Bats use them for flying, whales use them for swimming, birds use them also for flying, and cats and dogs use them for walking. So same bones, different functions. And this tells us that we all came from a common ancestor. Okay, analogous structures are next. Those are same function, different structure. So this shows that in the same environment, animals can come up with different ways of using the environment to their advantage. So for example, we talked about butterflies and bats. They both fly, but they have very different types of wing structure. So bats have bone and tissue, and butterflies have chitin. So they both evolved to fly in the same environment, but they have different structures for that flight. And then last but not least, vestigial structures. We talked about snakes that have little useless legs. Remember, vestigial structures are really small, usually very reduced, and they are pretty much useless. Some vestigial structures that humans have are wisdom teeth. We used to use those teeth for grinding tough plant material. We don't really eat that plant material anymore. We eat softer plants now, so we don't need those teeth. And usually what happens is they go unused and then they get infected and then they harm us. So that is why we remove those teeth. Tailbones, we do not have tails anymore, but we still have the bones for them. And the last thing you need to remember is cladograms. So remember that they start at the bottom, you start with the past, and you go up to the present, and the organisms listed at the top are the present day living organisms. The points where the lines come together are going to represent common ancestors, and remember that traits are listed with dashes on the cladogram as you go up. Remember that every trait applies to every organism above that dash. So that is how you read a cladogram. I think that is it. So remember that your test is open note, and use your notes, use your old quizzes, use your notebook. That's totally fine with me. I would highly, highly, highly recommend that you not use the internet. Obviously you can, but it's gonna give you some answers that are either going to confuse you or they're going to just be wrong. So I would stick to everything that we have from class. And please let me know if you have any problems with the Google form. I will keep my email up and running all day. So I miss you guys. I hope you guys have a great weekend and get some rest. Bye.